Good morning, everyone. So we'll start the next chapter and actually the final chapter of this course, which is the regulation in eukaryotes. Regulation in eukaryotes is much more complex, and there are several layers of regulation. Okay, and that is because of the complexity of the organisms itself. So as we will learn in the next slides or in the next class, simplest thing is this: we have one organism, and the organism has so many different types of cells, cell types with different functions, and yet they are all actually composed of the same genome, right? And then it would give rise to different types of cells. How is that possible, or what makes it happen like that? That means you will need so many levels of levels of complexity in terms of regulation. So we will have a few examples, maybe like transcriptional regulation in eukaryotes, like very similar to that of almost similar to that of prokaryotic transcriptional regulation, where different proteins act on the promoter and so on. The important things are about epigenetic regulation. That is the core of this lesson. But at the same time, you, as second year students, you may not have enough background or idea to understand these things, but we will still go ahead and try to touch on the foundations or the fundamentals of regulation in eukaryotes. In the next semester, I think there is for you, I guess there is an elective that is called as stem cell biology, fundamentals of stem cell biology. I take that course as well, and the majority of the focus, at least one whole unit, is dedicated for regulation in eukaryotes and actually epigenetics. And you will probably then understand how big and influential or important epigenetics is in eukaryotes. Okay, let's start, and then we'll go ahead slowly. Before going in, I just wanted to, because I had it in the last year, so I will just talk it anyway. Define intelligence. And this is also in light of your end sem exam, right? We are trying to teach you the depths of whatever is in the textbook, at least, covering the whole complex phenomenon. And it's like empowering you with tools. And then in the evaluation part, and we'll also try to train you about how to solve problems, right? And the exam is this. It's not necessarily, we will give you an exam in a question where you will have to use the tools that you're gaining from this course and solve the answers, right? That is intelligence. I am not interested in testing your memory. Not that memory is very unimportant or something. Memory is very important because everything is memory, basically. If you have learned algebra, it is still memory. Memory of the principles may not be the answers, right? So as an individual, the important thing is you growing intelligent every day. Every action that you take is to improve your intelligence. And the intelligence is learnability, or the ability to learn is the first thing. And the ability to learn will start with language and your interest, motivation, being in the right place, being surrounded by community where they are interested in learning as well. And then as a scientist, you should, the principal, in my view, the principal point of any scientist is to come up with questions, right? to come up with biological questions that nobody is interested in, or they don't know, or they are unaware of. And not just raising the questions or finding a question, it's also the ability to solve the questions or solve the problems that come up. These three things are the important things: ability to learn, ability to recognize problems, and ability to solve problems. And the third, I would also actually add the fourth point, that is ability to communicate. Ability to communicate your findings. If I got a good idea, fantastic idea, it's flawless and it'll work forever, but I don't know how to communicate it, then there is no value to the idea, right? So these things, I think you would want to reflect on yourselves and try to shape your growth, and you orient yourselves and your work towards these things. Am I improving in my abilities of learning? Am I able to recognize the problems? Do I have the tools to solve the problems? That's intelligence. Okay. And we also have discussed this slide. We have seen it before. And I actually use this slide in prokaryotic, or just to explain or introduce gene regulation. But in eukaryotes, this is much more relevant, because you have several steps: initiation of replication, exon splicing, passage through nuclear membrane, and stability of mRNA, the rate of protein synthesis, and these post-translational modifications. All these steps are regulated. What is the objective of regulation? Again, I think we have to come back to that question: is what is the objective of regulation? To be fit for the context, right? To be fit for the context, in the sense, it is basically ability to adapt. What terms is the adaptation? Adaptation in terms of phenotypes. Phenotypes or properties or characteristics, traits, you can call them any way you want to. And how can you change phenotype? You can only change phenotype by changing the proteome. How can you change the proteome? You can only change proteome by changing transcriptome. Right? So eventually, this is all regulation will happen at these levels: transcriptome, proteome, and so on. And that will result in changes in phenotypes. Those phenotypes are fit for that context. When I say context, it means the environment. Environment is not stable, isn't it? You have an organism here, and the environment is, assume this is representative of the environment. This is now fine, but after a while, the environment does not remain the same. Over a period of time, change is constant. And if the individual that is surviving in that environment should change according to the environment, changes in the environment, only then they will survive, otherwise perish. And that is why regulation is very important, right? It allows the adaptation of a cell or a living or of an organism to its surroundings. Okay. So as in bacteria, I mean, transcriptional regulation is the simplest or most predominant of all regulations that you will find across the organisms. In bacteria, we have seen about the promoter, and you may have an operator region. Sometimes you can call it regulatory sequence based on whether an activator binds or a repressor binds. And in yeast, which is a unicellular eukaryotic organism, you have some more additional elements of regulation to fine-tune the regulation of these genes. If you see in humans or other organisms, you find that the regulatory elements are huge. Even you may have even downstream of the transcriptional start site. You have operator regions or regulatory sequences, and you also have some enhancers. You'll also have insulators and so on. They're actually very complex. And that's one of the reasons why we do not know the complete extent or all the fine details of eukaryotic gene regulation. Some operons or sorry, some genes, regulation of some genes is simple, and we understand them quite well. But some of the other ones we do not know as much. But still, we will try to know try to understand the general principles of how their regulation usually is. So GAL4, I will just put it this way. There is galactose operon in Saccharomyces cerevisiae, or the yeast, and it has an upstream activating sequence called UAS, upstream activating here it is, for galactose. So whenever galactose is there, it should be produced. And for that, it has a protein called as GAL4. GAL4 is a protein which has two domains, similar to that of many repressors that we have discussed. Here is a domain that you're looking at. You have two domains. One is a DNA binding domain. You call it as BD, binding domain, or DNA binding domain. And the other one is called as AD, that is activation domain. So when there is need for expression of these gene of GAL1, then GAL4 binds to the binding site that is near the UASG, and it will, once it binds, and this activation domain is about transcriptional activation domain. So it will allow the binding of the RNA polymerase to bind and then initiate efficient transcription from GAL1. So presence of the GAL4 transcriptional regulator will increase the 1,000-fold increase in transcription of GAL1. So it's simple, like what we have discussed in activators in prokaryotic gene regulation. So there are at least 5 categories of proteins. They are all DNA binding proteins. And like what I was describing for GAL4, DNA binding domain. There are 4 different, at least 4 well understood ones, and also 5 HMG. The first one is called as homeodomain here. It has 3 loops, or these are helices, that is representative. The arrow, if they are represented like that, they are beta strands. Alpha helix, and beta strands. If you remember the protein structure of proteins, you will find these things, and also loop. These are the connecting ones, are these parts of the loops. So here there are 3. I mean, don't mistake this as a ball. It's actually a loop, alpha helix as well. Okay, shown in perpendicular to this view, and that's why you're seeing it that way. So homeodomains consist of 3 alpha helices, 1, 2, 3, and they could be with other domains as well. And then their functions will be determined based on that. But this is how they would bind to the DNA. The other one is called as leucine, zinc-containing DNA binding domains, which has one alpha helix and 2 beta strands like this, and they coordinate a zinc atom, and which is coordinated by or bound by several side chains of the amino acids, and using which they will interact with the DNA. And that is zinc-containing DNA binding domains. The third one is leucine zipper motif, where those proteins bind as dimers. They bind something like this to the DNA. And then helix, loop, helix. Why do you call it as helix, loop, helix? It's a dimer as well. This is a helix, and you have a loop, and another helix like this. And they dimerize, and at the same time, they are interacting with the, as a dimer, they are interacting with the basis of the major groove, right? I hope you remember what is a major groove and minor groove. This is a minor groove, and this space is major groove. Okay, and similarly, there are also several proteins called as that belong to a huge family kind, HMG proteins, high mobility group. They usually interact with the AT-rich regions of the minor groove of DNA. So typical of eukaryotes, this is a general phenomenon. If you remember the transcriptional initiation of in eukaryotes, we know that majority of the transcription is carried out by RNA polymerase 2. RNA polymerase 1 and 3 are for tRNA, rRNA, and so on. Most of the mRNAs are transcribed by RNA polymerase 2. And we also learned about there are several general transcription factors, and there is a mediator complex, huge one, which is involved in vivo. When transcription must be carried out in vivo, you will need a complex array of proteins, and those proteins will modulate the amount of transcription that is happening from a particular gene. I hope you remember TF2D, and this is Tata binding protein, and EF2B, it will bind to the Tata box. In eukaryotes, you can call it as Tata box, which recruits the RNA polymerase 2, and there are also several other factors like activators. Activator is the protein which binds to a site called as enhancer, and that will allow or amplify the recruitment of RNA polymerase 2 and 2F2D. It can influence either of them or both of them to bind to the promoter elements and initiate transcription. Okay? So that is about enhancer. Enhancer is a part of the regulatory elements in the DNA that are bound by activator, which when it bounds, activator can recruit other proteins to influence or to activate transcription. This picture must be familiar to you, right? We have seen when we were learning about genome chromatin organization, we learned that in eukaryotes, the chromatin is wound around nucleosomes. And in nucleosomes, the nucleosomes consist of histones, several of them, and that is why they will become very important in this chapter. Histones, we also learned about histone tails, and we also learned that tails could be, at least in two major ways, they could be modified, post-translational modification. We discussed about methylation, and we also discussed about acetylation. Right? These two are important. Mostly, if transcription is occurring, the histone tails are typically acetylated at that region. Upon acetylation, there are changes to the chromatin structure, and that will allow the accessibility of for the transcriptional proteins or transcriptional factors to recognize the promoter elements and initiate transcription. Let's see in the first case in A. So first, there is an enhancer here, and to the enhancer, an activator binds. The enhancer, the activator has two domains. One domain could be having any of the DNA binding domain, okay, like we discussed about leucine zipper or HMG proteins and so on. So they have a binding domain, and they have another domain here like this. That domain determines the consequence of what is happening. Binding domain only goes and binds to certain sequences in the DNA. But what happens will be determined by the activation domain. What is it activating? In this example, it is binding to an histone acetyl transferase, or you would call it as HAT, histone acetyl transferase. What does histone acetyl transferase do? It will acetylate the histone tails. When it is acetylated, you will find that the chromatin is spread out like this as compared to the previous one. The previous one is much more closed up, and the DNA is not accessible by majority of the transcriptional factors in that location or locus. So upon acetylation, the chromatin is spread out a little bit. Now there is this promoter that is accessible by the transcriptional factors and mediator complex, RNA polymerase 2, and so on. And then you will have the transcription activate. Otherwise, the promoter here is actually hidden away in the tightly bound chromatin structures. Okay. There are some things. There is one another way is by here. So activator is bound to the enhancer that is present by using its DNA binding domain, and then the activation domain. Here the activation domain is bound to or it binds to chromatin remodeling complex. What does chromatin remodeling complex do? It changes the conformation. It's not adding any groups or removing any groups. There is no chemical alterations. It is rather physical alteration, in the sense that the structures, the nucleosomes could be slided away, or it's like a mechanical work basically. The promoter which was in here, because of the sliding, you will find that the sliding of nucleosomes or replacement of nucleosomes. There are several ways of how nucleosomes could be rearranged on DNA. Please remember this. Nucleosomes are DNA binding proteins. Here the activator is also a DNA binding protein. I want to bring this to your notice at this point so that you will remember these things. Histones and activator here, they both are DNA binding proteins. There is one major difference. Activator is sequence specific, whereas histone, it is sequence non-specific. It's not specific to any sequence. It is specific to DNA. When you say sequence specific, that means this activator should be able to recognize the bases that are present in the DNA, right? So they are somehow interacting either in the major groove or minor groove. They're interacting with the side groups or atoms of the nitrogenous bases. That is only when they will be able to recognize what sequence it is. Whereas histones, they do not have, they're non-specific sequence. They're not sequence specific, which means that majority of their interactions would be with the phosphate backbone and maybe the deoxyribose. And the nitrogenous bases very limited, probably in the minor groove, which does not have so much of interactions or discriminating interactions. So that is one thing you have to remember. So because the nucleosomes are not sequence specific, they can be slided through DNA like this. And so the point or essence of this slide is that there are two ways. One way is the chemical modification because the activation domain of the activator binds to histone acetyl transferase. Whereas in the second category, the activation domain of this activator has affinity to chromatin remodeling complex. So that in both ways you can have enhancement of transcription. Okay, in both ways, the objective is simple: to make the promoter element accessible to the transcriptional machinery. Okay. So this, when we talk about the histone modifications, there are several things, right? HAT, just now we spoke about it, histone acetyl transferase, which will acetylate the DNA, histone tails, because of which they become expanded. When they are expanded, you call it as euchromatin. When they are very tightly packed, then you call it as heterochromatin. Right? If you are doing the banding patterns of chromosome, karyotyping, you call that, you will have, if you must have learned in your +2 or somewhere, a chromosome, when you do karyotyping, you may have darkly stained regions and light stained regions, something like this. The darkly stained regions are called as heterochromatin, and the light ones are called as euchromatin. These are stained dark because they are densely packed, and that is why they are densely packed, which is in the form of heterochromatin. And there is transcription is very negligible in these cases. The transcription does not happen as much. In euchromatin or true chromatin is the one where it is much more loosely folded or it is expanded, because of which majority of the genes are accessible for transcription, and the transcription happens from those regions. So histone acetyl transferase converts heterochromatin to euchromatin, and so there is gene expression. And histone deacetylases, HDAC, converts or removes the acetyl group, because of which they form back the heterochromatin like this. Okay. Similarly, we also have methyl transferases. We learned about the histone tails can also be methylated. And this is histone methyl transferase, which will methylate. And with methylation, in some cases there is gene expression, and in some cases there is gene repression. But as opposed to acetylation of histone tails, majority of the times acetylation is associated with increased gene expression, whereas methylation depends on the context and which histone is methylated, and which position of it is methylated. So when you say H3K4, I'm trying to make you recollect what you have already learned, which would mean it is H3 histone and lysine residue at position number 4, right? And so in that case, methylation of these residues will allow gene expression or transcription to occur. Whereas the same histone, H3K9, is methylated or K27 is methylated, then repression occurs in those areas. So it all depends on, there will also be multiple other factors, but you can remember some things, some general principle like acetylation is typically associated with gene expression, whereas methylation, in some instances, it is it is associated with transcription, and in some cases, it is associated with repression. There is also something like this, where the RNA polymerase will bind or kind of start the initiation, right? It is organized on these things. I hope you remember downstream promoter elements, starter box, initiator region, and initiation happens there, and it stops. Right? You remember promoter escape in prokaryotes and also in eukaryotes we have learned. And promoter escape is dependent on the phosphorylation of the CTD of polymerase 2, one of the subunits. So initiation, it's like in the first instance, it is loaded and kept. And that means it is because the serine 5 of the tail region is phosphorylated. But transcription does not initiate until the serine 2 is also phosphorylated. So that is also kind of regulation. The protein that is responsible, TEFB, PTFB kinase, if it is not around, then you wouldn't have transcription initiated. The mRNA should be produced, That is when you will say gene is expressed. Just initiation is insufficient, right? So although initiation happens, it is paused or prevented from going on to the next level or the elongation stage. So that is another level of regulations measure some genes can have. And there are also complexes called as RSC complex, that is remodel the structure of chromatin complexes that are what we have spoken about. Gal4 has the regulatory elements which are recognized by the Gal4 for Gal1. And when there are nucleosomes, there is a RSC remodel chromatin structure, which kinds of moves across the upstream of the RNA polymerase, and it uses ATP or kind of remodels that area or the locus, so that the nucleosomes are not an hindrance for transcription to occur. Okay. And in today's class, if you understood what I'm going to tell you about this, then it is good enough. You understand many things. And also remember, as I was telling you, if you were to take the stem cell biology course, fundamentals of stem cell biology, this is a recurring, majority of it will be focused on eukaryotic gene regulation at a higher level. In that course, we will come to this as well. I want to talk about loops and insulators. These are some looping we already learned a little bit, but insulators is something new. So what can happen is, say, enhancer is the place where activator binds. And the distance between enhancer and promoter could be huge. They can be very far off in distances. Unless there is a looping here, I mean, this one should bend like this, only then the activators are actually in proximity with the promoter, and then only they can facilitate the transcriptional initiation from this promoter, right? So DNA should be able to loop, and for that looping, you might require certain proteins sometimes to allow the physical proximity of these activators with the transcriptional regulatory elements. And there is a protein, there are regions which you call as insulators, to which something can bind, and a protein can bind, sequence-specific protein can bind. And if the protein is bound by insulator, the enhancer now cannot reach this promoter, and therefore the transcription from this particular gene will be off. In many eukaryotes, you will find complex genetic organization. Here is a promoter 1, and I will call this as promoter 2. And you have one enhancer. The same enhancer could be activating promoter 1 or promoter 2. It cannot activate the transcription of both at the same time, because physical proximity is important. In that sense, it will only be able to interact with one of the promoters. In that case, the presence or absence of insulator can decide which promoter is activated by the enhancer. So you can think of it like this. Either promoter 1 is active or promoter 2 is active, but both will not be active at the same time. Which one should be active, either 1 or 2, will be determined by the presence of these proteins on the insulator. Because if insulator is present, what will happen? It will hinder the looping of DNA. So although the activator is bound to the enhancer, because it cannot interact with the promoter, transcription is off. So in the case of C, promoter, if insulator is present, then This enhancer cannot loop to interact with promoter 1. Therefore, it will only go and activate promoter 2. But if this protein that is bound to insulator is not present, then the enhancer or the activator and enhancer can interact with promoter 1. And then you can see the regulation of the genes and which one is transcribed, one or two is active, is dependent on this protein that is binding to the insulator region. There are also several other genetic organizations. Here is a promoter, there is an insulator, there is an enhancer here, and there is also another enhancer. Either enhancer 1 or enhancer 2 will activate promoter here. Okay? These are general models of trying to describe the genetic organization and the influence of insulators on influence of insulators on the outcomes, whether transcription happens or not. In case of D, promoter, there is only one promoter here. It could be activated by either enhancer or activator bound to enhancer 2 or by activator bound to enhancer 1. Which one will do the job will again depend on the proteins bound to the insulator region. Okay, these are general points that is saying. One of the proteins is CTCF. Sorry. There are also several other proteins. That is, sorry, I thought I had it. Okay. There are also several regions called as locus control regions. Locus is a part of a gene, and this is one locus, you can say. And when you say locus control gene, that means locus control region, which means that if you either activate it or repress it, all these genes will also at a time. It's not like one gene, one regulation type, right? There are several genes. All of them either will be expressed or repressed at the same time. And that is mediated by certain regions that are called as LCR, or locus control regions. If there is repression here, all the cluster of these genes would also be repressed, something like globin genes in this. Okay, and in mouse also you have beta globin locus. They are also under the control of locus regions. There is even bigger one, which is called as global control region. It's present with several genes. That means this whole big part of a chromosome could be regulated by because it is having an influence much larger than LCR. Okay. So I'm not going to discuss about the consequences of these genes and what happens. I'm only describing that this is how parts of chromosomes can come under one regulation. If you're talking about immunoglobulin genes and so on, I mean, I will wait for you to finish immunology. Probably it's in the next semester. When you read that, you will also get better understanding. Otherwise, we will never be able to finish this course. Okay. And the activators, when they are activating, they have certain mode of action, in the sense that one of it is cooperative binding. I will describe those cooperative binding. And cooperative means binding of one will enhance the binding of another one, right? That is cooperative, inducing the binding of other protein to the activator region. How they are activating something. So activator binds, they're allowing the binding of other proteins, maybe other transcriptional factor, for example. In B, they could have the binding of A and B could influence the binding of another factor. And then there is going to be activation of transcription. The others are in indirect effects, like in C and D, where you're sliding the DNA, because A is bound. A is recruiting a modifier. The modifier is making the promoter, that is, which otherwise was not accessible, but now it is accessible because the chromatin has been modified by the modifier. In D, you have site A is there. A binding of a protein on site A will activate the, or will allow the sliding of the nucleosome a little bit, and because of which there will be, now the other part is also accessible and B can bind. These are general principles, not specific examples. Okay, whatever we are learning about today. Apart from these, we have learned about insulators, additional activators. We have learned locus control regions. We have learned global control regions, and how the activation. We have learned about types of DNA binding proteins also. Yes, and then we will skip this, and I want to give some introduction for the next class. And I think I already showed you this picture before. All tissues in our body have come from one single cell, and that is zygote, right? And zygote will divide into several cells, all looking identical, and that stage is called as morula. That morula will then first time differentiate into two different cell types. The outside layer of trophoectoderm and the inside, that is called as ICM, or inner cell mass. So zygote is one cell type. Morula, all the cells are looking the same, which are morphologically similar to that of a zygote. But in the blastula stage or blastocyst stage, you have two different types of cells. That is the first time you're seeing two different types of cells. One is the outer layer, trophoectoderm, and the other one is inner cell mass. The genome content of them is same, but there are two different types of cells, and that is the first differentiation. I think it is literal meaning, that is, they were something until then, but now they are morphologically something else, right? That is, they differentiate, you can differentiate the cell types. And that from all our tissues in our body, we have about 210 cell types in our body. They all have arisen from inner cell mass, right? And they have so many different functions. And how does that happen? It is all in gene regulation. So let's look at this. A zygote, upon fertilization, you'll get a zygote, and that forms a blastocyst or blastula, which will then, that is where you will have this cell mass here is ICM. Then it will form a gastrula, and I hope you remember that there are three germ layers. That is ectoderm, mesoderm, and endoderm. And there is also, these three germ layers are called as primordial germ layers, PGL, or primordial germ layers. And there is also, okay, primordial germ layers are the ones, they give rise to somatoplasm. What is somatoplasm? That is, all our body cells are somatoplasm, except the germ cells. Germ cells are the gametes, the ones that are involved in reproduction. Every other cells in our body they belong to the somatoplasm. None of the traits or differences or mutations in the somatoplasm will be inherited by the daughters or the offspring. Whereas if there are mutations or changes in the germplasm, they will be inherited by the offspring. So primordial germ layers and primordial germ cells. From the primordial germ layers are ectoderm, mesoderm, and endoderm. And ectoderm will give rise to certain cell types of skin, neurons, and so on. And the mesoderm gives rise to several of those, including bones, muscles, and so on. Endoderm gives rise to mostly glandular type, including stomach cells, thyroid, lungs, and so on. And from the germ cells, either they will give rise to sperm or egg. Again, in the stem cell biology course is mostly dedicated to understanding the early embryology a little bit, or basics fundamentals of it, and then we talk about gene regulation, eukaryotic gene regulation, epigenetics mostly, and then we will focus on how the properties of stem cells, and then we'll learn more about how to use the stem cells to address current day problems or in regenerative medicine. But here is what we are trying to do now is to provide a foundation for your understanding, so that you will be able to remember a lot. You will be able to carry those things in the future. So I will talk about this slide and then I will stop, okay? And when we learned the lesson about chromatin organization, we have learned that there is several fold increase in compaction. All chromosomes, if they were put end to end like this from one cell, they would be approximately about 2 meters. And they are put into, all of them is put into one cell, or actually one nucleus, which is less than about 10 micrometers. That's a huge degree of compaction. And that is obtained by several layers of ordered compaction. And if you have compaction, what is the problem that we were discussing in that also? Compaction, if it is, there is a problem with expression, because compaction will hinder the accessibility of the regulatory proteins or even transcriptional machinery, and therefore it can impede the expression of genes that are present on chromosomes, right? So, well, but then still expression happens. That means not only that these chromosomes are compacted, but they are also accessible, which means that the chromatin is actually dynamic in structure, in the sense that it can open up and compact again in locations that should be expressed. So DNA, upon binding to nucleosomes, will form something like this that will further form loops or compact higher, and then you will have the chromatin fiber. That is the typical one that we actually see. And then you will have further compaction, and eventually, if it were about to undergo mitosis or meiosis, cell division, then during metaphase, this is the compaction they would have had, right? And as I said, even though they are compact and still expression is able to happen, that is because of several dynamic ways, like we have learned about remodeling proteins and so on. The foundations of epigenetics, or the fundamentals of epigenetics, will depend on these four methods or types. First one, or the better understood one, is DNA methylation. DNA can be methylated. Because of that methylation, some genes are expressed, and in some cases the genes are not expressed. It again depends on which region is methylated and which one is not. Similarly, histone modifications such as acetylation and methylation can also influence and chromatin organization and nuclear architecture. Try to have a look at the at C, cytosine. Cytosine can be methylated in the fifth position. How would you count the fifth position? 1, 2, 3, 4, 5, and 6. In the assignment, I have given you one uracil in one of the sequences, which could have been uh, because of demethylation of thymine. What is the difference between thymine and I'll follow the same uracil? If you had something like this, say I'll count 1, 2, 3, 4, 5, 6. So if this is uracil, 5-methyl uracil is thymine, isn't it? And by any chemical means or so, this methyl group is lost. In DNA, you only have thymine, you do not have uracil. In the assignment, I gave you uracil, which would mean that demethylation of a thymine that might have resulted in uracil. And about repair, you should be able to say about uracil deglycosylase, uracil glycosylase, which is base excision repair, right? And then this would have been eliminated, and then uracil, the sequence would be repaired by what is present on the opposite strand or the complementary strand. So in this case, I also want you to remember that there are several GC islands, you call them CPG islands. C, P is for phosphate bond between them, phosphorester bond, and then G, guanine, gorgonosine, so on. So the cytosine could be methylated on the fifth carbon. That is, it will result in the formation of 5-methyl cytosine. Okay, so just remember this 5-methyl cytosine. Try to have a look at the chemistry, and then we will discuss further in the next class. Okay, I have uploaded an old end semester question paper. Let me stop the recording and I'll